just make one comment on the technology that's going on with nets. It's not dormant at all. And I heard you say that you have to replace 100 million nets a year, and that you got to reduce the pressure that's now being on the shoulders of the nets, so to speak. But there are new materials that are being worked on that will extend the life of the nets, make them stronger, and I just want to make sure that you know that that's not something that's just sitting there. There are developments.
Yeah, I mean, we have representatives from MMD and MBI here today. But there's another product development partnership at the University of Liverpool, the Innovative Vector Control Consortium, that's doing some of that work, and there's others who are working on that as well, working on new tools for nets as well. Yeah.
I was wondering how quickly, once resistance develops in the field with bed nets, we're able to detect that, and if there are implications for whether the best strategy is to go with a scheme that allows quick detection and react to that versus sort of planned rotations instead. If you have any thoughts on that.
There's been an awful lot of emphasis all the way from WHO to different working groups, and I know the CDC and the U.S. government, PMI folks, are looking at resistance both on the drug side as well as on the insecticide side. Detecting it is huge, so part of our whole capacity building plan is to look at developing centers of excellence or centers of response in regions and or countries so that they have an indigenous organic way of testing surveillance. Many of the countries it's very sophisticated, others it's non-existent. So it is a huge issue. It's being addressed, like Charlie said, this is not a new concept. There are a lot of very smart people at the university level, at the technical level, and at the implementation level, looking at how best, first of all, to identify surveillance, and then, resistance, and then what do we do about it, both on the drug and the insecticide side.
And David, maybe you could add, WHO recently released the global plan for artemisinin resistance containment, and maybe you could talk just a little bit, tell people what that is and how that may relate to that and MFD's role as well.
Yeah, I think I just wanted to make a few comments on that in that line, because the GPAC, as it's known, was released by WHO in January, really is an acknowledgement of the fact that there is an emerging problem in Southeast Asia right now, and we are seeing the emergence of artemisinin resistance. We don't know how quickly that will evolve and exactly where it will evolve, and so really the GPAC, it focused on a multi-pronged approach to addressing malaria as rapidly as possible to try and effectively either diminish or delay the emergence of resistance, but I think we have to treat it as an inevitability. And that includes the initiatives that we've been talking about today across the board, the development of new drugs, but I think one of the tools that's lacking currently is that despite the widespread use of artemisinins, we don't fully understand exactly how they work. And one of the issues associated with this is therefore there's no obvious marker for resistance. So in order to actually determine whether or not you're seeing reduced susceptibility to these drugs and classes, you have to do clinical studies, and that's not, and so these are clinical studies looking at the effectiveness of the drug in patients, and this is not a particularly rapid way of approaching it, and it's not really amenable to global surveillance. So what we do need is a far better understanding of the molecular basis for resistance, ideally to identify markers so there can be really widespread surveillance. So I think that's one of the key areas that beyond all of the very positive things that are happening, there are still some basic scientific gaps here.
Maybe while we're just to fill in the perspective of new tools, maybe you can also talk a bit about WHO's recommendation about the increased use of rapid diagnostic tests. 20 years ago, USAID supported PATH to develop one of the first immunochromatographic strip tests for malaria, which we transferred to an Indian manufacturer, is now one of the most widely used rapid diagnostic tests. one of the challenges is as we get further and further progress, success with control, fewer and fewer people with fever actually have malaria. So maybe you just talk a little bit about that.
I think that that this is, you know, we've mentioned some scientific gaps. I think one of the very positive things is there are rapid tests available for malaria. And this has a number of advantages. I think firstly, it allows you to far better target the use of drugs. This can have an impact on resistance. But equally, it has an economic impact. And this is a very positive thing. So, you know, one of the challenges right now is affordability and access to drugs. A lot of progress is being made, but a huge amount of progress can be made simply by the fact that you may have to use less of them if you use effective and cheap rapid diagnostic tests. So I think that's very positive. However, the use of those tests clearly needs to be scaled up because it's not universal. The other component is that if you're treating a febrile child for malaria and they don't have it, then there's another underlying condition that isn't being addressed, and that places them at significant risk of whatever that particular pathogen or illness is. So it would lead to better management of children. So the use of rapid diagnostic tests can have both a significant impact in terms of better use of drug, economic impact, but also health outcome in children.
I want to underline that last comment. Ray mentioned a minute ago that while malaria is taking on an infectious disease, it really is a maternal and child health tool. And underlying what David said, I think that's key as we look at ways to leverage malaria prevention and treatment and how it then dovetails into other health outcomes. This notion of perhaps 60% of the kids with a fever not testing positive for malaria means it begs the question for better health treatment. And so that's how we in the U.S. government are dovetailing very nicely and effectively with our maternal and child health programs.
Other questions from the audience? Yeah.
Coming back to the drug issue. Coming back to the drug issue, most drugs are developed as single agents. Is there scope within this, especially with the urgency, to develop early combination trials? So you may not necessarily optimize one particular lead, but you may begin very early to look at good combinations.
That's an extremely good point, and that's something that we're working on right now. We mentioned the compound OZ439, so that's now in phase 2A development. So it's early clinical development. It's a very promising compound, but there's a tendency to partner this drug with some of the older established agents, and to some extent that would be going down the wrong path. You know, it may not be an either or, but one of the options we definitely need to explore, and that we are right now, this is next on our radar screen, is there are other potential companion drugs that are also in development. And if we're to get timely advances of the magnitude that we want, we need to be looking at, and we will be looking at, partnering drugs like OZ439 with other development compounds at an earlier stage. So that's, you've definitely hit the target there. That's exactly where our R&D is taking us next.
Christian, maybe I could ask you a follow-up question to your comments. You mentioned the decision-making framework in preparation for a hopeful launch of the first malaria vaccine in 2015, and you also mentioned that the promising recent phase 2 study, follow-on study results. What are you hearing from African countries about their decision-making around a first-generation malaria vaccine that may only be 50 or 60% effective and not, you know, like other vaccines where we have an impression at least that they're, you know, one immunization and that's it for, 95 or more percent effectiveness?
David, that's a very important question. But maybe just to we've got other vaccines that are widely used today that are also partially effective. Pneumococcal vaccine, Rota are vaccines that don't have 100%, are more around the 60% mark, and they deliver very good results where they are widely used. So basically, that's already a very strong indicator that coverage, in fact, and uptake has to be very important. The first comment that we get from countries is always, can we get it faster? more than, in fact, a comment on the level of efficacy. And it's always to explain that we need to understand and have a full package of understanding the impact of the vaccine. That's why we're doing the phase 3 and all the studies to understand what impact the vaccine will have on the population. And maybe if I can give you an indication, in one of the phase 2 trials that was not statistically significant like experts were doing, but we had severe cases of malaria. In a group, it was a total group of more than 500 children that were immunized. In a group that received the control group, we had 9 dead due to severe malaria. In a group that received the vaccine, it was 1 only. So that was already a very strong indicator for us. And those elements are going, we know that malaria is very important in terms of comorbidities, in terms of morbidities linked to anemia. So those are the data that we're going to see. And basically it's not seen as, I would say, not seen as too important a problem by the African leaders.
One question maybe for both David and Christian is that I think we heard from Admiral Ziemer and Ray that money's going to be tight for the next couple of years, and we're going to have to work hard to sustain support for malaria control as well as research and development. There's been a lot of discussion within the Global Health Technologies Coalition more broadly about the role of innovative financing. Things like the advanced market commitment, different kinds of scenarios where donors might only have to, quote, pay for success, to create incentives for the pharmaceutical industry to invest more of its resources. Maybe either or both of you could just comment a bit on innovative financing mechanisms, what role they might play in bringing more resources into the research and development space, especially in this time of tight budgets.
Maybe I can start because we've seen one very interesting financing play, in fact, recently for the pneumococcal vaccine, where in fact came in to lead to 2 important agreements between, in fact, Gavi and 2 suppliers that committed a certain number of billion of doses. That one doesn't work.
Having trouble at the back.
Okay, so now basically that the advanced market commitment has worked and is in place for the pneumococcal vaccine, so we could envision that eventually for the next generation of malaria vaccine, it could be eventually used to give some incentive to large pharma and other industrialists that would be interested in investing. An important concern that we have is certainly when looking at Gavi, because Gavi is an important body to finance malaria, the vaccines, and we see Gavi having at the moment a shortfall, and it's very important to support all the initiatives that are going to help, in fact, bridging that gap and filling the additional requirements, because next in line in the 2 or 3 vaccine is the malaria vaccine, which would be successful. I think that we certainly need to attract in the field of malaria vaccine, it's not exactly an innovative financing, but we've been able to work and have a very good partnership with GSK. And last year, maybe you've not seen, but we've signed an important beginning of a collaboration agreement with Merck. And for us, it's very important to keep on attracting to that research large pharma, because it's important that they come and share, in fact, the cost of development as part of their CSR and corporate social responsibility of industry. And I think that that's pretty important to have that type of collaborations.